Okay, the final speaker before the break is Doxa from Crete Aquarium. She is a biologist with a master's degree in marine ecosystems, and she just 3 months ago started her PhD with the University of Crete. Since 2005 and the opening of Crete Aquarium, she's been an aquarist. And her presentation today is feeding behavior and food selection of the fasted marine gastropod in their experimental aquarium. Thank you, Krisa.
Buenos dias, good morning to all of you. I'm going to talk about the feeding behavior and the food selection of the giant triton Charonia tritonis variegata. This work was fully carried out in the aquarium of Crete, and it was a part of my master thesis. A few words about Charonia tritonis variegata for those of you who are not familiar. Charonia tritonis variegata is one of the biggest and most beautiful gastropods, and it's belonging to the family of the Ranelidae. As far as its geographical distribution is concerned, it is present in the Mediterranean Sea and in the Central Atlantic Ocean, but absent in the Black Sea, and it inhabits rocky bottoms, in the infralittoral zone and coral reefs. So, why are we interested in studying Charonia tritonis variegata? First of all, because it is an endangered species. Due to its overfishing, mostly for the trade of its shell, the populations have declined. Another reason is its feeding specificity, since it's an animal that is consuming sea stars, which until recently was supposed to be a dead end of the food chain. Charonia tritonis variegata is a close relative of the Indo-Pacific subspecies Charonia tritonis tritonis, which is a major predator of the coral-eating sea star Acanthaster planki. By mastering the life cycle of Charonia tritonis variegata, we hope that we will be able to contribute to the coral reef's conservation. A few things about the significance of feeding behavior from a biological point of view. Feeding is a factor that affects all the physiological functions of an organism in all the developmental stages. From an ecological point of view, feeding preference on an animal and subsequently its trophic position within a community gives us basic information about the ecosystem organization, which is very useful for ecological purposes like biodiversity management and biomagnification and biomanipulation for ecological restoration. The aim of the present study was to describe the feeding behavior and define the food acceptability of Charonia tritonis variegata upon 5 different kinds of food. These were a mix of sea stars, Luidia sansi and Astropectera asiacus, fish species, boops, the squid Nototondarus loani, the mussels Mytilus edulis, and the shrimps Parapeneus longirostris. Now let's pass to the materials and methods. Our experimental population was consisted in 47 individuals of Charonia tritonis variegata collected from the inshore littoral zone of Crete in Greece, and they were maintained in 150 and 300 liter aquariums at a constant temperature of 24 degrees of Celsius under an artificial photoperiod of 13 hours light and 11 hours dark. In order to be able to identify the individuals, we used the Dremel to mark the shells, so each one of the shells has a number indicating the individual. During their maintenance in these aquaria, they were fed upon sea stars, sea urchins, sea cucumbers, and fish. Now, the experimental procedure, we saw the food panel bit before, we used a mixture of sea stars. The reason of this mixturing was because we did not have enough quantities of each species to provide it separately. So we prefer to use in all experiments this mixture. And the other 4 types of food is feeds that can be easily found in the market in quite reasonable prices. So in the experimental conditions, we used 3 cylindrical tanks of 500-liter capacity with coarse gravel bed. The water was remained standing without air supplied during the trials in order to avoid the scent mixing and, if possible, not to confuse the animals. The temperature was stable at 25.5 degrees. Oxygen saturation was always greater than 9%. We kept the same photoperiod as the maintenance conditions, 13 hours of light and 11 hours of dark, and we used 15 grams of each food of each species per trial. 3 trials were conducted for each animal in a black cylindrical tanks, like the one you can see in this picture. After 1, 2, and 3 weeks of fasting, each individual was placed in the center of the tank, and the 5 different kinds of food were equally separated at its periphery. So here you can see the animal, here are the shrimps, here is the 2 parts of the sea stars, here is the squid, here is the fish, and here is our mussels. We recorded the time needed for each animal to sense the presence of food and start searching for it, the time needed to approach it, and the path it followed until they reached the food of its preference. Here you see an individual reaching part of squid and extending its proboscis to taste it. Each trial was stopped at this point when each individual was tasting the food before consuming it. In the 95% of the trials, the tritons started consuming the first food that they tried, but in the resting 5%, they tried one food, they didn't like it, so they changed their ways, searching for another one. For the statistical analysis, we used the analysis of variance, and we analyzed the differences with the Takei's test. So now let's pass to our results. As far as feeding behavior is concerned, the time needed to have a response to food was defined as the time between the placement of an animal in the tank and the time that the tentacles appeared. So we measured that time, and also we measured, which was varied between 5 seconds and 36 minutes with a mean value of 5.6 minutes. And we also measured the time needed to approach the food, which varied between 16.26 minutes and, which was, the mean value was 16.26 minutes. Well, in this diagram, you can see the mean time of response to food and of approaching it after 1, 2, and 3 weeks of fasting. Although there were no statistically significant differences, you can see that the time was from the first trial to the other 2 was reduced. In this series of pictures, you will see the way the Charonia tritonis variegata responds to food and searching for it. Here you see an individual placed in its aquarium and the presence of food, a piece of starfish. The first thing to appear is the tentacle of the individual, while the food is still not widely open. Then slowly it starts extending its food and searching inside the tank for the food. You see that it moves its tentacles in order to locate the food, and when it approaches it, it uses its proboscis. You see it's this part here. Here it can be more easily seen in order to consume it. First it tries it, and then if it likes it, it constrains it with the use of its food. Now, as food acceptability is concerned, here you can see the results of the trials. The highest percentages were 28 and 27% for the sea star and the shrimps, followed by fish with a 21%, squid with 16%, and finally mussels with only an 8%. The difference in lowercase letters represent a statistically significant difference, so as we can see, the choice for the sea star and the shrimps differs from the choice for the mussels. Passing to our conclusions. As far as feeding behavior of Charonia tritonis variegata is concerned, these animals, like all gastropods, uses its tentacles to orientate towards food. There are a lot of mechanisms developed in carnivore gastropods to consume their prey. Charonia tritonis variegata possesses an extendable proboscis for food consumption, and they use their food to restrain the food. No statistically significant difference occurred between 1, 2, and 3 weeks of fasting, probably because they are animals with a very low metabolic rate, so perhaps a greater period of fasting is necessary to differentiate the results. As far as food acceptability is concerned, we saw that all the 5 kinds of food were accepted. It was the first record, at least from what we know from bibliography, that they accepted squid, shrimps, and mussels. We found a higher selection rate for sea stars and shrimps, and a very low selection for mussels. In the end, I would like to acknowledge the director of the Aquarium of Crete, Mr. Papadakis, the aquariology department and the technical department for their help, and of course the fishermen, which were the basic key for this research, because they brought us all these individuals of Charonia tritonis variegata. Thank you for your attention.
Thank you, Chrissa. Are there any questions? There's one here at the front.
Daniel Arbeit from the Haus des Meeres in Vienna again. I imagine that you also spent some time looking for this, Charonia in the wild, I think. Or did you just rely on the fishermen finding?
No, we rely on the fishermen. Mm-hmm. We do not do it by ourselves. So, a few individuals were collected by people of the Hellenic Center of Marine Research. So we know where they found them, and some others were collected by divers that do it as a hobby, and they bring the individuals to us. So we have a few information about their natural habitat. But the majority of these were collected by fishermen, by nets, etc.
What would be interesting for me is if there is a difference in sea star abundance in areas where you find the Charonia and in areas where you don't find them anymore. Do you have an idea? Are there more sea star in areas where there's no Charonia anymore?
Unfortunately, I do not have an idea about this thing.
Thank you.
Okay, thank you very much.